Welcome to presentation 2 of 4 of Meiosis for the Alberta Science 30 curriculum, Unit 2, or the Biology unit. So in Meiosis 1, you had a diploid cell. These chromatids are lined up at the pole in tetrads. The tetrads are separated to produce 2 haploid cells, or 2 one-end cells. These sister chromatids, or these chromatids, DNA is further separated into chromosomes, and again you result in 4 haploid cells at the end of meiosis 1. So in meiosis 1, you have the nucleus starting to break down in prophase. The chromatin is being condensed into, and the DNA is paired into these things called chromatids, or sister chromatids. These are paired in these structures called tetrads because they have 1, 2, 3, 4 chromatids there. In prophase 1, crossing over occurs, so you get some mixing of DNA between these 2 pieces. Then in their tetrads, they are arranged on this metaphase plate. The spindle fibers pull the chromatids apart, and cell division occurs. A nuclear envelope is reformed, and the chromatids again uncoil and become chromatin. In meiosis 2, you have the chromatids condensing again. You have the nuclear membrane breaking down. You have the chromatids being lined up on the metaphase plate. In this case, it is in a single row. These chromatids are separated into chromosomes in anaphase 2, and in telophase 2 you have 1, 2, 3, 4 non-identical daughter cells being formed through the process of cytokinesis. A tetrad is a structure formed when replicating homologous chromosomes. When they come together during prophase 1, a tetrad consists of 2 replicated chromosomes, or 4 chromatids. So 2 chromosomes, or 1, 2, 3, 4 chromatids. The centromere is the center point that connects the 2 sister chromatids together. The whole structure is called a replicated chromosome. Chromatids are chromosomes that are still attached to their sister strand via a centromere. When the centromere eventually splits and the chromatids are free of their sister chromatid, they are then called chromosomes. Humans have 23 pairs of chromosomes. Chromosomes 1 through 22 are called autosomes. Chromosome number 23 is called a sex chromosome because they determine the sex of the individual. The sex chromosomes are X and Y. In this case, this individual has 2 X chromosomes, so is a female. In males, this process is called spermatogenesis. In females, this process is called oogenesis. It takes place in the testes for males and the ovaries for females. The spermatogonium, or the parent cell, the 2N cell, will produce 4 equal-sized cells called spermatids, with each having an equal amount of cytoplasmic organelles and cytoplasm. In oogenesis, meiosis 1 is unequal. One cell is much smaller than the other and becomes what is known as a polar body. In meiosis 2, there is another unequal cell division with another polar body being formed, and one egg being formed that has most of the cytoplasmic organelles and cytoplasm. In spermatogenesis, you have a spermatogonium. In meiosis 1, you produce 2 one-end cells. In meiosis 2, you produce 4 one-end cells from the 2 one-end cells that were produced in meiosis 1 to result in 4 sperm. In oogenesis, you start with an ogonium. You have an unequal cell division with one cell getting the majority of the cytoplasm and organelles and one cell being very much smaller than the other. Again, you have an unequal cell division, and the result is 4 small cells called polar bodies and 1 large cell called an egg cell. The egg, or otid, gets most of the cytoplasm. The polar bodies die and are absorbed by the body of the mother. Again, here's another picture of spermatogenesis. You have the spermatogonium, which is diploid. You have this primary spermatocyte. You have the autosomes pairing in these tetrads. In meiosis 1, you have these autosomes separating into these 2 cells, the secondary spermatocytes, which are haploid, and then you have further separation to result in 4 haploid spermatids, so 1, 2, 3, 4, which will mature into a sperm. Meiosis produces sperm cells. This occurs in the testes, in the seminiferous tubules. So if you look at the seminiferous tubules, you will see these diploid cells turning into these things called primary spermatocytes, and then resulting in meiosis or mitosis produces primary spermatocytes, which through the process of meiosis 1 and meiosis 2 produce sperm cells that eventually mature and are released. What are the structure of the sperm? You have this head, which consists of the acrosome and the nucleus, designed to get through the egg, get through the protective coating on the egg and the nucleus containing the DNA. You have a tail and all of this mitochondria designed to propel it through the woman to fertilize the egg. In oogenesis, you have an ogonium, which produces a primary oocyte, which is a diploid cell. Through the processes of meiosis 1 and meiosis 2, you produce a secondary oocyte, which is haploid, and 4 haploid cells, one of which is an ovum or egg, and 3 polar bodies which die, which mature into an egg and may eventually get fertilized and produce a baby. So again, spermatogenesis produces a sperm, oogenesis produces an egg. The difference is 4 sperm are produced by the males for every 1 egg produced by the female. Fertilization occurs when sperm meets egg, and the very first cell is called a zygote. The zygote first becomes an embryo, then a fetus, and then when it is born, eventually a child. This process of forming an embryo, a fetus, and a child is primarily done through the process of mitosis or cell division. Males make approximately 10 to the 9 sperm cells every day. Females are born with approximately 400,000 egg cells. 1,000 mature every month, but only 1 leaves the ovary.